Welcome.
So, of course, welcome to all of you here. Nice to see a lot of familiar faces, but also a bunch of new faces. That's always good. And of course, we have a bunch of people who are connecting online. Um, so Modi and I were talking earlier, and usually it's the other way around, right? Usually the speaker is on Zoom and the audience is sitting somewhere, and that's how we are also used to it. It's going to be a bit unusual where you have not half, some fraction of the audience here, a large fraction that's going to be from. So, by way of introduction, MST is a program that we conceived of 2 years ago or so with the idea of reaching out to college teachers and to sort of aid them in how they can go about doing their job of teaching a bit better. Now, I see there's a bunch of students here, so I guess you're all college students, BScs, MScs. So, I don't know if you know this, but a college teacher typically does not undergo any training before they are told to teach. Right? So in school, for example, you do BA or an MA, but a college teacher does research, does their MSc, maybe sometimes does a PhD. Then they join a college, then they are told, okay, go teach this class, and then they try to make the best of it. Now, of course, from your end, you know firsthand how well that goes. But if we want that to improve, the right thing to do is to figure out how we can fix the car while it is running. Right? That's what you're kind of doing. You can't take the teachers away for 6 months and say, okay, I will train you. Then you go teach. Who will teach in those 6 months? So as stuff is happening, our idea is to do interventions, to have a bunch of workshops where we do a bunch of different things. One thing that we'll do is we'll try to bring in new pedagogical techniques and give the teachers exposure to that. The second thing we'll try to do is we'll try to bring together experienced teachers and younger teachers. So a college teacher is usually a smart person. If they've been teaching for 10, 15 years and so on, they have their own set of tools and techniques in how to deal with difficult topics. They know how to talk about their own subject, whether it is bio or math or whatever. So we also want to build this community where you have younger teachers and older teachers all come together on one platform. So for that, we have this MSD program through which we try to go to all of the different districts in Maharashtra and try to eventually address every science teacher that exists in Maharashtra. Now, that is a tall goal, but we've made reasonably good strides in trying to do that through a model that, if you're curious, you can come talk to me or Asim later. One of the things that we'd like to do when we do this teacher training is the fact that how do you motivate a teacher who day in, day out is teaching the same course and has to do the same things? How do we make sure that we can pull them out of that and introduce them to different ideas, not because they will not hear it otherwise, but because that teacher needs an excuse to pull away a couple of hours, right? With the mindset that, okay, now I'm going to do something new. An easy excuse is the programs that we do. So that is the idea of having a webinar like this. We have the resources because we've been doing this for a while to actually manage to catch exceptional people to come give talks. So in that sense, we're very fortunate. They agree to us. For example, it may be difficult for Kamuri to go visit 10 colleges in Maharashtra. She may not have the time to do that. But if we manage to take care of facilitating it, then her job is easy. She can reach more people and more people can then take part. That's the idea. The aim of the webinar is, of course, that you hear some really novel things. They force you to think. And given that many of you have a background in science, one doesn't have to sell the fact that you should think. And that's all that you should do. Think a lot. So the hope is that this triggers thought in you It's interactive, as you can clearly tell. So the hope is that you do stuff interactively. This also goes out to the people on Zoom. The hope is, you manage to find interactions that, you manage to find activities that you can do there. First, somebody will guide you as to what you should do if you're attending this from home or office or wherever. But for the people here, I think it definitely will help if you are, you dial up your interactiveness to a little more than what you would usually do. Because a lot of the audience is online, I think they will also help if you sitting here take part more than what you would usually do. Like not like a classroom, right? Where you find the place in the back. So we move those girls up there. Good. So that's the idea. I think enough by way of introduction. I'm going to hand it over to Kamudi now or Asif. And so he will introduce Kamudi to us. And I hope you have a fun time. The topic looks absolutely fascinating.
Wonderful. Thank you so much, Saurabh. So along with Saurabh, there is Chakrapani, who is also heading this project with us. We are always, blessed and encouraged by both of them. When they come here, they talk to us. So thank you once again. I'm going to read out. This is not very often I'm going to read out community info, because he has done so many things. Starting with the latest. So let us welcome Dr. Kamudi Bhave. She is the co-founder of BioM. It's a consultancy service focused on working with small startups, where she supports large and small molecular drug discovery, development, and company diagnostics. She also serves as a senior scientific advisor for Cancer Patient Lab, which is a non-profit organization devoted to empowering cancer patients and their caregivers in charting their own paths in the complex arena of cancer treatment. Kamudi received a doctorate degree in public health with dissertation and research focused on machine learning based on causal Bayesian networks for unraveling distinct roles of NRF1 transcription, these are factors from management. But we will ask her to discuss that later. So these are transition factors in advanced brain cancers. So see the diverse background and what she is applying. She has published her work in multiple peer-reviewed journals and also presented at conferences. Kamudi has passion for learning and I like teaching also. So this is where we met close to 9-10 years back. Here at ISER Pune when we did a boot camp, which was on so she's also interested and she does conduct interdisciplinary and science workshops all over. This is mainly for participants of all ages that you see here and also online. Now this is something what Saurabh was also mentioning, that having such kind of a workshop where the meaning of multidisciplinarity or vertical education comes to be such kind of workshop. Of course, she loves teaching, facilitating, and co-learning in these events. She also likes to customize workshops that she designs, something what we are doing now. So let us welcome Kamudi and hand it over to her.
Thank you so much, Asim, for that kind introduction. And hello, everyone. Welcome to the workshop, as well as to the participants who are participating, I guess, from multiple different locations via Zoom. So the brief background about myself kind of spoke about the fact that I like teaching, but I like co-learning. So what I'm expecting all of us today here, whether you're in person or in a hybrid mode, to sort of learn together, okay? And I have on purpose chosen a topic which can be enjoyed or sort of explored by people from different backgrounds. So I just wanted to get a feel for people. Can you just, by raising your hands, just show me how many of you have a biology background? Okay, so a lot of biologists. How about physics? Anyone? Okay, we have, okay, great. Okay, and then chemistry. All right. Okay, so we have the traditional physics, chemistry, biology sort of represented here. And I mean, online, we'll take a poll later, kind of find out that too. But this particular topic, which is the biological self across space and time, I would argue applies to everyone, whether you're in the sciences or even not, right? And so we will use hopefully what is like a science-based scientific approach, but using multimodal learning. So you'll be using hands-on, like there's colorful blocks and materials on scattered on the tables. And so for those of you at home, one of the assignments was to kind of gather a little bit of your random materials at home, because we will be doing some activities. All right. So when we say biological cell across us, before we even begin the actual crux of the talk, the workshop, as Saurabh mentioned, there's a reason why these sort of workshops are happening. And the need of the hour is to create the next generation of scientists, researchers, teachers, who will bridge those gaps that currently exist in current research. So we've seen multiple, decades of having this traditional sort of breakdown, physics, chemistry, biology, at the school level, at the undergraduate level. If there are any PhD students here, or anyone who's PhD and beyond, they might realize that those divisions as physics, chemistry, and biology really don't hold up much when you're on your own trying to research one specific topic, right? Then you really have to pull from multiple angles to sort of help you propel your research. So the thought here in bringing these multiple angles to our subject at hand is to sort of get your brains in that space already of picking up cues from multiple sources, verifying the sources, actually understanding the scientific background or the scientific environment in which these sources have been created, okay? And then using concepts from multiple areas to tackle challenges that you're facing in your own learning, research, or teaching. Which is why all of you, whether at home or at these tables, you will see an assignment that has been placed in front of each of you. And essentially, there are two things in that assignment. The first bullet point being that in the back of your mind, as you're listening to me speak and as you're participating, think of something that really has been bugging you, okay? Something you understand, or maybe something in your research that you'd like to explore further. Just, there's a blank sheet of paper also kept for the participants here. At home, if you have a notebook or something, just jot it down. What is it? What is it that you've been sort of struggling with that's, really something that you would like to learn more about? So I'll give you a couple of seconds to just do that, to just jot that down, okay? And another thing, as you're jotting that down, another thing that is placed on the tables, it's for sharing. There's one copy per table. It's a handout about something called Minkowski space. Yeah? Okay. So I think the online participants will need that specific document. Okay, perfect. So take a few moments to read that document. You can jot down one thing that's been on your mind that you're kind of curious about. It doesn't have to be in the subjects that you're learning at school or college, okay? It can be some random question. You're walking on the street and why is there, how could traffic be diverted better in Pune or something like that? Any question, just jot it down. Or, yeah, what makes weather patterns? I mean, anything, just a question. throughout this workshop, I'll be giving you sort of 2, 3 activities in parallel to do, so that any person can start on whichever activity first. If it gets too much or it's too much instructions, please raise your hand and give me that feedback. Okay. The third activity to do at each table or at home is to take some of the material in front of you and just place a few objects. I'll say place 3 objects in the center of the table or in front of you if you're at home. 3 random objects from the supplies you've been given. And you can place them in any arrangement. Take your time. Remember, just 3 as a group. Each group is only placing 3 objects in the center. That's it. And now, your definition of object is something you have to agree on as a table. What do you, as a table group or at home, what do you mean at your table? You make the rules. What do you define as an object? And then place the objects in the center. All right. If your table is done with all 3 given activities, just raise your hands. I want to get like how many people are done with all 3 activities given. Okay. All right. Now it's time for us to move on to something else. Okay. And trust me, there is a method to this madness. So you bring random things, but then first, and we'll be talking about concepts back in your, or they might link in your brain. I would like everyone in this room and online to take a moment to just take a deep breath, ground yourself in the spot you're seated, and then gently Close your eyes. Close your eyes. Everybody, close your eyes. Hang with me here. Close your eyes. And imagine that you are on the deciding committee. Okay? The funds grant deciding committee to decide whether this specific topic is funded or not. The topic at hand is a new research department or group to be started to be studying the biological cell, space and time. And you're in position of authority, capability to make the decision of whether to allocate a large amount of funding for this project. In your mind's eye, imagine exactly how you visualize. Where are you seated when you're making this decision? Indoor space, outdoor space. Okay, do you have a chair, table? Just visualize that space in which you're seated as you're making this decision. Okay? Everybody who's in this room can feel free to participate in this activity. Visualize the space well. You can even visualize the color of the furniture, the room, whether there's a window, a door. Are you alone in the office or is it a group of you, a committee that's making the decision together? Take that time to visualize it all. How are you feeling as you're going to make this big decision? You need a lot of information to be able to decide whether something is worth the funds or not. You're thinking about what sources, scientific. How do you even decide what is science and what's not science, right? But I want you to focus on visualizing this location. Your mind's eye, where are you seated? Is it in Pune? Is it in some other city? In India or some other country? Think about it. Your mind's eye, visualize it. And now please gently open your eyes and immediately take out the blank sheet of paper and I want you to jot down something. You can just add some bullet points as a list. Is first thing, what was the distance between two pieces of furniture or yourself and a piece of furniture? Space that you visualized in your mind's eye. Estimate it. What was the distance? Or you can estimate the distance between door. That space that you saw in your mind's eye. Give me one distance between any two things that you saw in your mind's eye. Secondly, estimate the area of the room you are sitting in. Take a moment to estimate the area of the room that you are seated in. Okay? And the third thing I would like you to estimate is that how long this activity took. So how long were we visualizing in our mind's eye? These three things I would like you to jot. Would anyone like to volunteer what they came up with? I see some people have kind of finished the writing process. So it's a fun. You don't have to be tense that, oh, my math, no, estimations may be way off. That's okay. It's just for something else. So anyone can share? Anyone else wants to share who visualize yourself sitting in some space? Five minutes. Someone who estimated less than 1 meter of a distance between the desk and themselves. And for 5 minutes. Yes. Anyone else would like to share something else? Okay. No one was out there. Okay. Anyone else wants to have any other opinion? Anyone online? I don't know if anyone wants to has contributed online. Does anyone have any input there at home?
They were not able to hear
Oh, okay.
because of this mic. So if you are speaking into the mic, just
Speak louder. Are they able to hear me?
if you could use this one, which is a better one.
Perfect.
It
That's
should, so right.
Can
Okay. She's saying she can hear you, but not the participants.
I see? Okay.
So everybody should speak in this.
All right. So now I guess online people can hear me better and all of you can hear me. Okay. So the general consensus, I mean, people were saying 2-3 minutes, 4 minutes, kind of like that. So by the watch on this cell phone, it was 1 minute that we were with eyes. Okay. Now different people had different like alag andaaz the of what how much space or how much distances were there. Okay. Just keep that in mind. And now I'm going to talk for a bit with slides. So all right. So today we are here to learn about or think about biological space, biological self across space and time. The first question that we can ask is space absolute or relational? Is time absolute or relational? Is self absolute or relational? These questions may not make any sense before we define the concepts we're talking about, okay? The three concepts we're talking about are self, right? Yeah. Self and we restrict it to biological cells. Space and time. So, can anyone take a crack at what is cell? What do we mean when we say cell? Anyone? Raise your hands if you ever actively thought about this already. What do we mean when we say cell? Okay, we have a few people. Okay. What do we mean when we say time? How is time defined? If anyone wants to quickly check on their phone a definition that's common, the definition of time, the standard definition, feel free to check on your phone and share with me. What, if you just had that question, ask Dr. Google or Google, all knowing Google. How do you define time? So, what's the English dictionary definition of time? And maybe someone else can do the same thing for space. How is space defined? How do you define space? Anyone raise your hand if you have the definition. You just have to read it out of your phone. Excellent. Okay. So should I give them the mic or? Okay.
You can repeat something.
Because it's stronger. Okay. So yeah. So I think we'll come with the mic and.
The measured or measurable period during which an action, process, or condition exists or continues, or we can say duration.
So in the definition of time, the word comes duration, right? The duration wouldn't, can you repeat and I'll try to repeat that.
The measured or measurable period during which an action, process, or condition exists or continues.
Okay. The measurable period. So you see the period, duration, these words. This is what is called a circular definition, right? Okay. Was anyone able to find the definition for space on their phone, on Wikipedia or Google or somewhere? Okay. You care to share?
A space, a place or an area that is empty or not used.
A place or an area that is empty or not used, right? Area. What is this? Again, circular. Now the last one being cell. Let's check if Google has any answers. Cell. So just to orient all of you, today's topic is such that it's kind of, there's a philosophical aspect and then there's a very hardcore scientific aspect. And believe it or not, there are hardcore applications, especially in today's day and age. Okay, there are many applications of trying to explore these areas, which I'll be getting to those later. But there's a big problem in actually finally defining these concepts. That's okay. We're still going to ask the question, with whatever working knowledge we have of space and time and cell, even though we don't have a formal definition that everybody can agree upon, or that is not a circular argument or circular definition, as the human species, we haven't come up with that yet, right? Such a definition of space, time, or cell. Nevertheless, all of us are walking around thinking we know what is time, right? Anyone asks, do you know time? Yeah, of course. Or do you know what time is? Yeah, I know what time it is right now. Yeah. Same with space. You kind of already were able to see in centimeter areas. Same with cell. You think you know self and we're talking with self. Okay. Now, when I say, okay, any of you who are interested after this workshop can look up on YouTube and online that there's been a debate for many centuries in the scientific community. Newton, the Newtonian view is that there is something as absolute space. If there is no matter existing, there is still space.
right? Fundamentally. Same with time. But many philosophers before him, and then Einstein as well, and modern science kind of agrees with Einstein's view, is that, for the most part, is that there's no such thing as absolute space or absolute time. It is only an emergent property of relationships between existing matter, essentially, or entities. Okay? That's a pretty difficult concept to kind of grasp. That there is actually no such thing. Maybe it's just a theoretical concept. Okay. So the reason some of you may be wondering, why are there just 3 dots on this whiteboard? And why did I ask you to put 3 objects in the center of your table? Now we are all existing on Earth. There's an atmosphere. So between those things, there's atmospheric air. There are molecules. We know that. But let's just do a thought experiment. What if all of us were just sitting in a vacuum as a thought experiment? Can't really happen. But what if the concept of vacuum means actually nothing is existing between the 3 objects that you placed? Okay. What does that even mean, right? Suppose our universe consisted of these 3 dots. How would you define or describe such a universe? Suppose it consisted of only 1 dot. That's easy. You don't have to do anything. That dot cannot relate to itself or anything like that. It's just 1, right? But if there are 3, how do you describe the 3? Exactly. You have to do that in relation to each other, right? Even looking at 3 dots, so look at 3 objects on your table, begin describing. If you have to describe all 3 as a group, you have to describe the relations. And in order to describe the relations for this particular activity, it's easy to see how you would have to bring in the theoretical concept of space, right? What about time? Is it necessary to use the concept of time when describing the 3 dots on this whiteboard? Anyone? Is it an unnecessary concept? Do you need it? Feel free to say something loudly, okay? How many of you think that the concept of time is necessary to describe the 3 dots on the whiteboard? Raise your hand really high if you think it's necessary, okay? All right, so we have about 50% of the room. That means that's telling me that the other 50% disagrees. So those of you who refuse to raise your hand, you are also saying something, you're making a statement, right? Those of you who did not raise your hand, can you please say why you don't think time is so necessary? And both arguments are legitimate in this. Anyone? Why? You don't need time to describe this, right? But they're already there.
It will depend on the question that you're asking about.
Excellent. Okay.
If you're asking questions like, how did this arrangement? Yeah, it will depend on the question that you're asking. So if you are interested in knowing how did this arrangement, come up, or what may happen with it eventually, etc. So then,
Thank you for sharing that. That's an excellent point. I hope everybody got that. It depends on the type of scientific questions you would wish to ask, right? Remember that. Remember that as you learn the material you're learning, or if you're trying to teach a class, or if you're embarking on a research project, actually. Okay. Oh, so we are talking about in all 3 concepts, are they absolute? Meaning, does a self exist that's outside of this physical body or not? Does space exist outside of any matter or not? Does time exist, on its own independently? Okay. Now I'm going to run through some different various concepts from biology. Okay.
Here you have on the y-axis, you have space or volume occupied, and you have time on the x-axis, right? And these are biological scales of organization, right? In school and college, everyone learns, yeah, there's molecules can organize into cells, like tissues from organisms, organisms organize into communities, and those communities organize themselves into ecosystems. Yeah, and then the planet, you go larger, right? If you're a physicist or someone like planet, then planets, then the solar system, and go on up to galaxies, universe. Okay, fine. Now, whenever we use these scales, us humans with our brains use these concepts of space and time as we learn science, as we teach science, we just take it for granted without ever asking the fundamental question. We just ponder upon it. That's good. Okay. Now we're going to operate at that level. So now we're going to ask some questions. Oh, humans are able to conceptualize these things. What about plants? Do plants sense space and time? I see some nodding heads because I see some biologists, I see some people who are familiar with this. How do plants sense space and time is another question you could ask. Okay, if they do, then how do they do this? What about bacteria? What about fungi? What about viruses? Viruses being on the boundary of life, right? Whether it's a question, are they living or non-living? Okay, do they sense space and time? Oh, and by the way, if I forgot to mention this at the beginning of the workshop, my role here is literally to just introduce you to all these questions that should get you to have your thinking caps on. That's it. Okay. So, right. So there is evidence, as biologists know, there is evidence suggesting that there is a shared understanding of space-time by multiple organisms of the same species. And I have listed 4 different examples. Yes. Don't worry, all of you may not be familiar with what is bacterial quorum sensing. We're going to look at that. There's migration of butterflies, birds, right? There's synchronized nesting, meaning they go to a particular place and lay eggs, or they have their young ones. And then there's simple synchronized swimming of fish. If you haven't seen it, it's very hypnotic, right? So to give you a break from my voice, we will be watching some of those short clips to kind of bring us in awe of how nature works. Like there is evidence suggesting this understanding. Okay, so play this here.
So this behavior of bacteria working in groups, we now call that quorum sensing. And so the way it works is bacteria need to understand times when they're alone and times when they're in communities so that they can behave differently when they're alone and when they're in the group. And so the way that they do that, they can't see and they can't really talk. And so the way bacteria know when they're alone or when they're with neighbors is that they make and release small molecules. So as the bacteria grow and double in number, they're all releasing these molecules into the environment. So the more cells there are, the more of the molecule there is. And when that molecule hits a particular threshold amount, the bacteria detect it and they infer from that detection event that they must have neighbors around. And so in unison, all of the bacteria change their behavior together. So that's quorum sensing. It's a chemical communication mechanism that allows bacteria to orchestrate group behaviors.
So was that video clear about quorum sensing in bacteria. Okay.
The largest insect migration in the world ends each The largest insect migration in the world ends each year in Michoacan, Mexico. Cold winters are deadly to these beautiful butterflies. So when the chill of winter descends on the northern half of their range across the United States and Canada, they take flight en masse in search of a warmer winter home. An estimated 100 million monarch pass the cold months of the winter hibernation period in the towering trees of this beautifully protected nature reserve in western Mexico. On their incredible journey, the butterflies travel an average of 2,500 miles, relying on air currents and thermal drafts to carry them long distances. The monarch is the only butterfly known to migrate the same way that birds do. At night, when the temperature drops, they cluster together in tight groups to conserve heat and energy. This survival tactic produces some of the most stunning formations across the insect world.
In this video, is actually in shot in India. This you're seeing. In this particular video, you're seeing the nesting turtles who travel long distances to all gather on the beach together at the same time. So, similar to the question on the board about the butterflies, is how do these turtles track space and time, right? These are active research questions right now. Even researchers are not 100% sure of how this happens in diverse species. This understanding of sort of space and time is happening in the case of the butterflies. It's clearly happening in the case of the bacteria. That's through chemical mechanisms of the world. It's really happening in the case of the turtles. And of course in humans, right? Yeah. Can I forward to? Yeah. So they lay eggs at that, so you get the idea basically. So next, yeah. And then this final video is about synchronized swimming of fish, if you've ever seen a school of fish. That's on a smaller time scale, right? We have things happening on large distances, large time scales, and then even in smaller time scales, we see synchronized events. Synchronized swimming for fish, we have synchronized events for cells even in the body, right? How is this happening? So, all right. You biologists are probably familiar with this. I don't know how to make the top thing disappear, but it's okay. There's something called circadian clocks. How many of you have studied this and are very aware? Show me where your hand. Yeah, a lot of you are aware. So I'm going to go a little bit fast through this part. But essentially, these are endogenous, they're self-sustaining, time-keeping systems that occur. They're molecular at molecular level. And they exist in most examined biological forms. So whatever has been examined today, there is some mechanism or the other of timekeeping. But one of the interesting things is that even though they have been examined in multiple organisms. You look at this last point here, the clock genes show little conservation across species. However, the mechanisms are similar. Okay, that's interesting. And these sort of clocks, molecular clocks, they have an intrinsic period length of about 24 hours under constant conditions. And anyone want to just share, shout out why 24 hours is so special? Anyone? You studied this? Why is 24 hours so special? Make a guess. What does it relate to? Why, Exactly. Thank you very much. It's because of dark and light, the Earth's rotation. So biology is related to Earth sciences, in that sense, right? Okay. Environmental cues, such as light, temperature, act as time givers, and those are based on the Earth's rotation. So that's an important point to remember. Okay. Now here I'm showing you a basic molecular clock that has been studied in cyanobacteria. It's called the Chi ABC oscillator. It's one of the more well-studied sort of and beautiful, elegant systems in these cyanobacteria. It's single cell organisms, right? But look at this. If you look at the four corners of this diagram, essentially it's three proteins, the Chi A, Chi B, and Chi C, which are interacting with each other and causing molecular changes. So here you have this Chi C, and the Chi C protein being the most important in this kind of a group, but you need all of them. All right, you need at least two of the three, but I'm getting that. So you have this Chi C protein, which has these serine threonine residues that can be phosphorylated, and you have Chi B and Chi A that can interact with Chi C. And depending on those interactions, there'll be changes in the phosphorylation states, and there'll be changes in what other molecules are binding to Chi C. And this particular molecular structure is something that the cell can recognize and keep time in that manner, right? So this is just one of the many such clocks that exist in organisms. So circadian rhythms in mammals, like humans, how do we tell time? Now this is focused mainly on time, right? The clock proteins, the molecules are existing within the cells, each of the cells, right? One mechanism in mammals is where the BMAL1 and clock, these two particular proteins, heterodimerize. So both different proteins come together and sit at the E box of the DNA, and they can activate circadian regulated genes. So what does that mean? So these are two factors that are sitting on top of the DNA and actually governing which proteins are turned on and off at the cell. So there's groups of proteins that are turned on, groups of proteins that are turned off, right? So it's a beautiful mechanism that's happening. And then there's feedback loops. So if you have too much of something, there's a molecule which will go and shut it down. So biology, it's full of feedback loops. Okay. So then in humans, all this is, remember, it's happening in all different cells of our body, in different organs and tissues and everything. But all of that gets integrated. Those signals are integrated in something called the suprachiasmatic nucleus in the brain. And that happens to be the central pacemaker. So you need kind of like the boss of the organization, telling the entire organism, like, how is this time working, right? Because if your liver is working on a different time and then the stomach at a different time, like, it's going to be chaotic. So you have this, I'll give you a moment to read it, but essentially through light, through other various different input signals, it decides that clock for the entire human organism. And this diagram is just showing you. See, you have here on this side some external signals, like the light outside, what food you're intaking, what are the, if you're doing exercise, if you're socializing, all of these factors will affect how your own body is sensing time. Internal signatures includes temperature, and then the signaling molecules, hormones, all of that, right? And nervous system signals. So we began the class with, I was asking you to estimate the time it took to visualize, right? Just think about this. If someone's really anxious, like had a lot of coffee and
they might think, oh my God, it's 4 hours past and why isn't she telling me to open my eyes? If someone is super calm, they might think, oh, less than a minute past, it feels so good. But so these things actually affect the way your own body and your brain is sensing time. Okay, fine. So it's relational in that sense. Now, something very interesting is that these circadian rhythms, okay, we all need these to function. We all need these to walk around to have an idea of the kind of world we are operating in. For our survival, it's important, right? But why would a bear that's hibernating in deep sleep in winter, still researchers have recently observed that the energy production continues to follow a daily pattern, right? Even as they sleep for months. So now my question to all of you is, why would a bear in hibernation need circadian rhythm? Anyone wants to think about it? And yeah.
Probably to self-regulate.
Okay. Yeah. Can you expand on that a little more? You're on the right track. To self-regulate, yeah. Anyone wants to add?
It has to make sure that all the processes are still going on inside it.
Correct, because can't completely shut down everything. That could be one point. What about
Bears? The bear's hibernation is dependent on the season and on external factors. So it has to be in touch with them to understand when it has to end with the hibernation.
excellent point, right? The bear, very good. So what was said is that the bear has to sense when to end this process. So it's necessary. Good. And another thing could be, for example, what if there's a sudden danger, right? So that's why the systems have to be on. Okay, fine. Now we're going to watch another video which talks about how our brains can process sense and time, space and time. Yeah, absolutely. Oh, we love questions. Okay. Pause. I think I paused. Yeah.
So while you were discussing hibernation, I was thinking about when people go into a coma or something like that, what happens to the circadian rhythms there?
That is an excellent question. I don't know the answer to. Does anyone else in the room know? That's what I meant by co-learning. See, this is the beauty of these workshops. Thank you. What is your name?
Purnima.
Purnima, thank you so much for asking that question. Does anybody know the answer? I don't actually. That's a great question. I'm guessing they would. I'm guessing that yes. Because many people who go into many years of coma still come out of it. So my guess, educated guess, is that yes, the circadian rhythms are still going. That's an excellent question. Maybe homework, someone can look it up or someone online can contribute if they know, right? So, and this is exactly why these workshops are important. So any of you, any such questions that come up, either jot them down, share them. Yeah. So we're going to watch this now.
Decoding dimensions, how the brain maps time and space. Time and space, the very fabric of our existence, the realms within which all our memories, thoughts, and future visions are constructed. But have you ever stopped to ponder how our brains actually perceive these dimensions? New research sheds light on this phenomenon. Researchers illuminated new pathways of understanding about the brain's interpretation of our world. Imagine the vast network of neurons inside your head, each playing its own special part in the grand symphony of your consciousness. Among these are what we now identify as place cells and grid cells, the brain's internal GPS. These cells are responsible for giving us a spatial awareness, allowing us to map our surroundings and understand our position within them. But what about time? The ever-elusive dimension that constantly moves forward, yet always seems to escape our grasp. Researchers discovered the existence of time cells, a distinct group of neurons that keep track of time. During specific tasks like navigating virtual environments, these time cells showed incredible adaptability, changing their behavior depending on the situation, all while place cells remained consistent in their operation. Think about watching a movie uninterrupted. As the story unfolds on screen, how does your brain keep track of time? Some unique neurons display periodic behavior, activating at regular intervals, acting as the brain's stopwatch. These neurons maintain their rhythm regardless of external influences, such as if the movie speeds up or slows down. So what does this all mean for us? This research has unlocked a profound understanding. The brain is not just a passive recipient of information, but actively decodes, interprets, and reconstructs our experiences. These discoveries highlight the incredible adaptability and intricacy of our brain's mechanisms, giving us insights into the biological basis of our cognitive maps. Essentially, our brains are designed to encode, navigate, and comprehend the dual dimensions of time and space. This newfound knowledge opens doors for further research, to delve deeper into the intricate dance of neurons and to better understand the symphony of our consciousness. So the next time you reminisce about the past or daydream about the future, just remember, your brain has already mapped that journey for you in both time and space. What do you think about this research? Drop us a comment below. Don't forget to like and subscribe for more neuroscience news updates.
All right. So I just wanted to bring participants here awareness of the fact that we have people online who are contributing, and some of them have sort of addressed the question you asked. And there's some people who contributed saying that since there's lots of consciousness in a coma, there's probably not that happening. Space and time, no sense of space and time there. Okay, but I'm going to leave this question to all of you to research further. Okay, so thank you to online participants for participating as well. Okay, so let's escape and slide. All right. Now, if you remember the first slide, the first slide had a bunch of questions on it, including how do plants sense space-time, if they do, and viruses as well, right? So now for plants, of course, in most organisms, there's those clock proteins, molecular proteins, but something else to keep in mind is that there is this aspect of resources, availability of resources. So light and temperature, maybe food, all of these are actually resources. These are the inputs that an organism is getting, right? And interacting with the environment. So plants need to be able to sort of regulate, or not, I should not say they need to. They do regulate how sugar is used. And that's the way in which they sort of sense time. So here I've just highlighted, it's a lot of writing and you can take your time and read it. But essentially they do this by this highlighted material at the bottom where there's a general shift happening from catabolism to anabolism, right? And so that's the key thing I wanted to bring here home in this slide is that organisms use resources, and the way they are actually using those resources, they adapt. And that plays a role in sensing time as well as space, right? Space is a resource. So different pathogens, so coming to viruses now, different pathogens target specific organs of the body, of the human body and other larger animals, right? To proliferate and make their home and spread, right? Now, different organs in the body have their own circadian mechanisms happening, right? Why do I say that? Because, say, even the clock gene, suppose the basic molecular mechanism of the clock is common for all the cells in the body. Still, liver cells have a different set of proteins that are being expressed, right? Stomach cells have a different set. Bones, muscles have different sets of proteins being expressed. And depending on this circadian rhythm, the patterns of the genes that are on and off, turned on and off, so the proteins that are being expressed or not, those are different for the different niches within the human body. Think about that. It's yes, the patterns change, but the subset of genes that are changing are different based on location in the body. And so pathogens that interact with those specific organ niches are interacting with different subsets of proteins at different times of day, at different times of year, etc. This is a non-trivial point, by the way, for those of you who are interested in research in biology. This, what I'm saying, is a non-trivial point because it can be a rich place in which to start asking some research questions, right? And on this particular, in this figure, the pathogens that are listed here, including viruses, some viruses, are known or have been shown in the literature to interact with circadian rhythms in their pathological niche. So, just wanted to say that here. And now, after this, so we kind of addressed, do humans sense space and do other lower organisms, we call them lower, I don't like that term actually. So, do plants sense space-time? Do viruses at the boundary of life? They say, well, we defined, well, no, we didn't define. We talked about cell, space, and time. What we didn't talk about is the word sense. What do you mean by when you sense space and time? That goes straight to the hard problem of consciousness, and we're not really going there today because of we don't have time to do that. Okay, but we're going to switch gears a little bit. But I want you to remember how we looked at the fact that plants and other organisms interact with their environment, and the way they are processing resources comes into picture about how they sense space and time. So again, we looked at this diagram at first, but these are biological scales. There's this particular paper that, and oh, by the way, disclaimer, all the papers that I've used as references here, none of them are my personal views. I'm just bringing you material organized in a specific manner, helping you facilitate your thinking, okay? So I will share these resources. We will be later, and then you can read and learn on your own. And one method I wanted to bring you home to is, you get a resource, right? You start up, there's one paper you find interesting, but then you have to look at that paper, where it came from, which group studied it, who else is studying that area, which other papers are have counter arguments, etc. This is a technique that has to be used in science, and so all of you people who are younger and start practicing early, so that it becomes habit, second nature. And for that reason, before this workshop, we shared a teaser yesterday. How many of you had a chance to read that particular teaser, that little write-up? Be honest. No. Okay. Well, if you did, good. Okay. If you've read it beforehand, that's extra bonus to you. Good. We're going to be talking about that next. But first, this particular paper, it's a vision paper from 2019. And it's put together by this University Corporation for Atmospheric Research from a particular university. I'm going to let you research which university and which organization I'm talking about here. There's not much about it, but they have many such conceptual vision papers on their website, okay? About large topics like this. Large topics like this one being, how do organisms deal with varying resources? So their view is a metabolic view, okay? Keep in mind that they're the research. So these are scientists that are interested in the environment, right? And their thesis is kind of geared towards how environmental conservation is going to be important, because as resources are dwindling, suppose, then organisms are going to shift their metabolism, and then there's going to be there could be harmful effects as well. But more importantly, they point us to something called, under this diagram, if you read this here, we need scale-independent hypotheses, okay? These three words mean a lot. Scale-independent hypothesis. We have biological scientists who work at the molecular level. We have a different set of research labs that focus on the cellular level or tissue, just translational biology, something like that. We have people working on the ecosystem. I mean, of course, more and more there's the new fields of molecular ecology and there's all these kinds of different things. But when you think, when you're a young researcher starting your project, think about this. What are the concepts, underlying themes that can cross scales of organization in biology, especially, right? Okay. So they deposit or their position is that there are resource types that exist across scales, okay? And the resource types that they list include free energy, this energy, right? Free energy, forms of energy storage, and information. So they're not even talking about air, water, food, like that. They're talking about something different here. Okay. So when you really boil that down to something, first principles, what resources and processes span the biological scales? You can say, in short, energy and information. It all boils down to energy and information, right? Whether it's at the cellular level or organs, and systems talking to each other in a body, or in an ecosystem, different species interact. Energy and information. So they go on further to say that exchange of energy, mass, and information at any given scale translates into a biological entity or unit that can be studied. Right? This is okay. So at each biological scale, there is an example of the form in which the information is exchanged. But nevertheless, the information is exchanged. That being the underlying thing. Now, at any given biological scale, whether you take molecule or you take ecosystem, right? The processes can be classified either as pertaining to energy conductance between two components of that system, or energy storage by individual components of that system. Okay. So this can help in interdisciplinary, transdisciplinary, within a subject, even within biology, for example, right? Say. Okay. So what is metabolism, right? Metabolism is essentially the processing of energy transformation. What's happening at that Krebs cycle, biochemistry, all that, everything? It's transformation of energy and it relays information, right? And it's happening by uptaking nutrients from the environment. So, yeah. So for this particular paper, this is a lot to read. I don't expect you to read that right now, but this paper will be shared with you later, and then you can look at their website and all the other papers they have as well, right? This is their goal is to unify life sciences under the theme of resource limitation, as I mentioned. It's a different way of looking at things or teaching things or learning things, right? And now, let's see what time it is. Okay. Now we're going to go to, I'll leave this on the screen. It could be a little distracting, but I'll leave it on the screen. This is a paper that was published late last year in Nature. Okay. And it has caused quite a bit of controversy, debate in the scientific community. So those of you who read the reading assignment prior are perhaps aware a little bit about that. Okay. Because the reading assignment that was given prior to the workshop was essentially an opinion piece, a summarization of all the back and forth in the scientific community that has happened ever since this paper was published. Now, this paper attempts to have a unifying theory which will allow cross-fertilization and research between evolution, so biology and physics, for example, right? Across disciplines. And there are some scientific problems or challenges that researchers have been faced with for many years. One of them being, how do you define life? So, what does it mean? How do you define life, right? That's why we all learn in biology textbooks. Viruses are on the boundary of, living and non-living. But what does that mean, right? Because it's a challenge. How would you define that? So this paper attempts to do that by something called assembly index. Okay. Now I'm going to escape out of this presentation for a moment. I probably need help to do that. And I'll bring up the PDF because I want your draw your attention to something. Okay, while we're waiting, this perfect. Actually, it came up. All right. Thank you. Okay, this is good. This is good. Okay. So here are the authors. You can see the list of authors here. And you already have the reading assignment with you that will point you to scientists with opposing views to these authors. Okay. Now, basically, I'll give you a moment to look at this. I don't know if this PDF can be shared on the WhatsApp or the. Okay, perfect. So all of you should have received the PDF on the WhatsApp as well. So those of you who are at home can open that up, the PDF, or if you want to do that on your phone, or if you want to look at the screen, that's fine. Just reading this abstract basically. Okay. I'll give you a couple of moments to just read the abstract. Activities. Okay. So now you've kind of read the abstract, whether on the screen or your phone or at home. Kind of a dense abstract. It takes some time to see what exactly are they saying then, right? Okay. Before you even read this abstract, in the beginning of the workshop, you were handed out a handout on Minkowski's space. In the middle of the table was placed Minkowski space. So take a look at that again. And essentially, Minkowski space is a way in which to represent space-time together, right? We usually have just space, three-dimensional space on the XYZ axis. But Minkowski space helps you to visualize space-time together. And now, before we tackle point number 2 in your workshop assignment, I'm going to write some questions on the whiteboard to sort of jog your brain and help you to answer this question, because this is a little bit more difficult than perhaps some of the questions I'll be writing on the whiteboard, okay?
Hopefully some fun questions. Let's see. Okay. And we're going to have a discussion on the questions that I write on the whiteboard. Oh, and online participants, please join in on these questions. Would they be able to see the questions as they get? Okay. Should I have this? Keep. Not visible, yeah. I realized that actually. I'll just. I can also just say the questions if that helps. So I'll just read out the 3 questions I posed. I think it's because we're liking them. The third question is, how would you describe yourself in Minkowski space? First one is, how would you depict, visualize, or describe ICER in Minkowski space? Next is, how would you depict this workshop that we're conducting here in Minkowski space? And then it's a tongue twister, no? And then the third one is, how would you describe yourself in Minkowski space? And then another one is those objects that you placed on the table. How would you describe those in Minkowski space? Something to think about. Yeah. Yes.
Okay, I'll repeat what I said right now. Okay, so for many biologists, this happens, and that you see a formula in a paper, research paper or somewhere, and you just the first reaction is it looks very ugly or very intimidating, right? And your mind blanks out. So when that happens, one technique is that this big monstrous looking formula exists here, let it be, okay? And then read the material around it. Put those things into concepts that make sense to you. Another technique, even if it's not a formula, but it's like that paper that I showed the abstract with, which is on the screen right now. A lot of dense language to understand. Then it feels like, oh, where do I even start? Forget it, I'll just do something else, right? Whenever you get that feeling, one good way to do it is, okay, fine, there's this big chunk of text, right? Let me first highlight the words I know. And does that make any sense? And then underline or highlight the words you don't know. Find out the meaning of those words. And then try to say it maybe in your own home language even. Right? So if you speak Marathi, Hindi, Malayalam, Telugu, whatever it is, try to say that in your own language, out loud. Really helps when understanding a concept. Okay. So one of our online participants contributed this idea that they can visualize it as 3 concentric spheres, maybe. Okay. Yourself, and then the outer one is the workshop, and then the outermost is ISA. See, that's conceptually easy to understand, right? You don't need any mathematical formulas, so don't worry. Okay, fine. But is anyone who's here available, willing to volunteer to draw any axes to this? You can be wrong. It's okay. Don't be afraid. Another thing I tell younger people who are entering science is don't be afraid to be wrong. Okay, you're going to be wrong for most of the times, right? Anyone who's in research or professors who are attending will agree that you're going to conduct experiments and you're going to be wrong most of the times, and you have to keep modifying. So, anyways, does anyone want to come up and try to draw axes based on the Minkowski information you have at hand? Absolutely. Thank you. Yes. You can take another one.
So I think this the same two axis. I think the 2 axes are important. The first is the space x component and y is time. So I think all these circles, they will be at the same level, I should say, that the time is same for all. And for the x component, the self is I think near about to this crossing point. Then Iser will lie outside here somewhere. And then the third, workshop will be the next, and Iser will be the outermost. And the cones, I think, they will look like this. This is self. This is the cone. Then this is the workshop. Now the most is I think so.
Okay, thank you very much. And so this is one representation, right? Based on the information you've been given. Your name, sorry. Rajita Kanikar has shared with us that she would like to visualize it like this based on the information. Now, our online participants, I'm so sorry, I'm not getting, I'm not able to verbalize everyone's names, but you're all very special and valuable to this workshop. So please continue to participate. So we have this view. Does anyone want to venture to see if there is any relationship between those spheres and the cones, or is it wrong to say that one could put it as spheres? Any of the physicists or the physics people here maybe wanna. Or I mean, if you look at that again, we're not going to get to the answers today, but these are concepts to struggle with. And I'd like to know if anyone wants to attempt, whether there's any relation. I mean, so we saw that one of the participants, while trying to draw axes to those spheres that were suggested, landed up kind of converting that into the conical forms. Okay. So this process of refining your conceptualization is something that is important when you are learning a new concept and even when you're teaching a new concept. Many times your students will come and challenge you also, right? You may have been thinking about something a certain way, but then you realize, oh, that doesn't explain this part of the issue at hand. And it is this very same process that is used when scientists are debating about scientific theories, the validity of scientific theories. So, this much time we took to struggle with this Minkowski space, think about it, and okay, fine. The key point being space and time can be visualized together, and it's a powerful concept which allows us to do other computational and other kind of operations on the entities that get represented by Minkowski space, and it's a known tool that is used, okay, by physicists, by mathematicians, right? Okay. So the reason I brought it up in a group that is predominantly biologists is to also for you to understand that the time has come for physicists and biologists and mathematicians to talk to each other and learn from each other. Because computational biology, it's just exploded if you pay attention to the field. And so probably a lot of you are going to be doing research in highly math-intensive, physics-intensive areas of biology. Okay, so it's something to get over the fear early is always good. Okay, and now having said that, we go back to this big nature paper, right? So I saw a few people, I'm not going to put you on the spot, but I saw a few people who said they did read the reading assignment before coming to the workshop. Does anyone want to volunteer the gist in 2-3 sentences, what they got, even online people, if anyone's asking?
There are a couple of comments for the earlier activity.
Oh, okay.
From So one, yeah, so one comment is the first diagram looks like a top view of the second. Yeah, so that's one comment. And then one person asked that while drawing XYZ axis, what scale can be used to the relationship? He's talking about the scale of the And he's the same person who contributed the sphere. Yeah, the sphere.
Yeah. Okay, so I'm going to tackle this scale question. Okay, that's very interesting because and let's tackle it conceptually, right? So we have a mixed age group here, right? So, like when I ask the question, how would you represent yourself in Minkowski space? Say someone, I think we have someone in school also here who tagged along with their mom or dad or someone. Maybe I saw that in the initial. I don't want to put it. Oh yeah. So, are you able to share how old you are? 13. Okay, so wow, we have maybe our youngest participant who is 13 years old. Okay, so you can imagine with a Minkowski space for someone who is a teenager or younger, is going to look very different from the Minkowski space diagram for someone who's 85 years old, suppose. So, what kind of scaling for the axes would you need to represent depends on your sample, your population, right? That's one concept. And bringing our example again, this workshop, participants, the workshop. So the participants themselves have a space-time existence. It's kind of creepy it's a little theory when you think about it. You have a space-time existence, not just an existence right now in space. And then again, it brings us back to the question, is space absolute? Is time absolute? Is the self absolute? Or is all of this just relational and happening because we're relating to each other and there's no such actual thing? That's philosophical questions outside the realm of science. Anyway, for now. But you can now visualize without focusing on the equations in that handout that the diagram for an individual in this workshop will look very different from Minkowski diagram compared to the diagram, say, for ICER, which has been around for now how many years? I don't know. When was ICER started? Anyone? This question. 2006, 2005, 6 time frame. Now it's been like what now? Almost 20 years maybe? Yeah, something like that. Yeah. So now, what about the ICER space though? And what is ICER? Is ICER the buildings? Or is ICER the people who are here right now? Or is it the people who've been alumni or past professors? Is it? So then how wide does that space stretch and that time? What is the scale? That's a very interesting question. We can just ponder on. Okay. Let's see. Yeah. So now I'm going to sort of try to give you an idea in short, okay, about this paper. And then we're going to leave this paper and there's another paper that we're going to give you to read optionally, which hits upon these concepts of self, space, and time. So the assembly theory paper basically uses a technique in which they have come up with something called, thank you, exactly that's where I wanted it, the assembly equation. Okay. Now I force you to look at equations. So in this paper, look at this equation, but the concept, okay. So let's see this pointer here. This is the equation here. But what is it exactly? It's a summation, right? And it's a summation over n, I is equal to 1, e naught to n, of what? But A is the assembly of the ensemble. So we're defining an ensemble of entities, whether they be biological or non-biological, okay? AI is the assembly index and NI is its copy number. So this assembly equation or assembly index is something which accounts for two components to any given object. Remember at the beginning of today's workshop, when I asked each table to place 3 objects in the center of your table. And I said as a group, decide what do you mean by object. Our conventional way of thinking about objects is what? What is it? An object. What is it? What's an object? Physical entity. Honestly, answer me this. Do we typically think of time as a component of any object when we describe it? Anything which is occupying space. We talk about space. We kind of ignore the time component of it. But the reason I bring you to this Minkowski space is there is a way in which you can include the time component in it. But even more interesting is this particular paper, where they are saying that time is embedded as information in any object. If you take a slice of time. Yeah. So I see some audio, I see some people who are aware of this paper, and it's a very interesting paper if you get to read it, okay? Because right now, suppose we were to take a photograph of this room or kind of describe everybody in this room at this moment, very moment, right? We account for the space we are occupying. We would probably describe the volume of this room. We would describe, the chairs in the living room. We will not account for the fact that each of us has lived before coming here, and we'll probably live after it. Yeah, okay, hopefully, yeah. So we've lived before coming here, so we all have a past. Now, this assembly theory is accounting for that past. In which way? Not like a historian, but like a historian, because they're saying any object can exist only if objects prior to it have existed. So any object that you see at a given point in time embeds its entire history in it as information. Does that make sense? Yeah? Okay, okay. How many of you know what is or can define what is a recursive phenomenon? What do you mean by the word recursive? Anybody? Recursive. I need two minds whether to write it on the board or what to do, but because we're limited with time now, we're nearing the end of the workshop. Okay. Can people hear me still with this? Yes? Online people can hear me? Okay. So actually, I'll just say it. It's easier instead of having you watch me write. Recursive is a procedure or a rule that is repeated. Does that make sense? A procedure or a rule that is repeated. So the crux of this paper is that anything that exists embeds information about the recursive elements that allowed it to exist. It tries to address that question we all have, that how come we see only a subset of all the possibilities that could exist in this universe or in general. Anything, in order for it to exist, has this long history of recursive elements. So in biology, let's take and these examples can be found in biology and physics. So so if you're studying, say you're studying the Western Ghats. Suppose you're a geologist studying the Western Ghats and you're studying rock formations and things. Then you will know about the sedimentary, igneous, and metamorphic type of rocks. So embedded in the rocks is their history, right? Okay. Also, in a different way, in chemistry, we see this all the time, right? If you have to have a DNA molecule, first you have to have those atoms that are going to form the molecule, right? And then in biological systems that we see on planet Earth, we mainly see, what do we see? Carbon, hydrogen, oxygen, nitrogen, maybe phosphorus, used in these. We don't see other elements being used in such large quantities to construct these biological entities. Why is that so? Because one is the properties of those atoms became conducive to creating molecules that became conducive, on and on, right? However, even that being said, there could have been so many biological entities that we don't see right now. Like, why am I not seeing a room full of some people having 6 eyes, some 8, some having 4 limbs, some having 8 limbs, some having 2 heads? Why don't we see that? A lot of powerful scientific questions can be asked when you ask why you're not seeing certain things. By studying what is missing, right? Why? Asking that question. How come? Or how does this work? Okay, so they propose that any entity can be described, one, by the number of recursive steps, the minimum number of recursive steps in other smaller entities that went into its formation, one. Secondly, by the number of copies of that particular entity. And the number of copies of any given entity. Just to bring it home, why are there so many humans on this planet? Because we're proliferating and we're able to. So a process of selection is happening in place. This process of selection they propose is happening, not they propose, they just kind of formulate it in a way in which we are able to quantify this. Okay, so that if you have a formula mathematically and you can quantify a certain factor which can be measured whether something is living or non-living, then you have a way of studying this. And what they observe is that there is a threshold above which all biological entities have. So this assembly index of all biological entities is much higher than the assembly index of, say, rocks. I'll allow you to read the paper on your own. Please, if you can read it, try to understand. Read the controversy. That's why I sent you the controversies of the debate first. Read that because it's interesting. Okay. And if you do, if those of you who did read the debate, were you able to see there's a video in that? Yes? Okay. So I won't spoil it for everyone, but watch that little video too. Did you watch the video that was embedded? No. The video, because you scroll, it feels like clickbait, maybe don't click on it. But there is a video in which they talk about the debate between Mandelbrot and Simon. This is in the older times of science. Okay, they're debating. And in scientific papers, they're actually calling each other out. I don't agree with what you're saying. I don't agree. And this goes on for multiple years. In today's day and age, it's going to happen like over Twitter or something. It's going to happen quick. And you see that with this paper, which was published last year in a prestigious journal like Nature, right? But there's scientists saying, oh, why did it get published in Nature? It doesn't deserve to. There's all kinds of material out there. Read and you'll see. So, okay. Last concept of today's workshop, before we bring it to a close, because of our time, time again, is, let's see if I can bring up the other paper I just wanted to. And both these will be shared with you, the PDFs, I think. Please. Yeah. I think you can just open it from here. Yeah, perfect. And yeah, finally, some of you may be wondering, we talked about time, we talked about space. What about self? So for self, you go to the interesting field of cognitive science, right? Identity, cognitive, yeah, exactly. How do we comprehend or understand what self means? Very interesting, right? So this particular paper, as you can see, is written by one person, and you can see his affiliations as well. And it's in Frontiers Journal. Frontiers of Psychology. Yeah, Frontiers in Psychology. So cognitive science, psychology now. And he talks about, I found this very interesting. That's why I wanted to bring it to your attention. It's a very novel, I thought, way of looking at it is that he's talking about the computational boundary of self. Okay. So this title itself I found intriguing. Just take a moment to just read the title and see if it makes any sense to you, right? Just the words in the title. Developmental bioelectricity drives multicellularity and scale-free cognition. Okay. So, this is a quote from 1950. See, of course, there's no question that a tree or an elephant is one individual. Like we all say, oh, look, there's an elephant, look, there's a tree, right? Okay. And we have a clear mental picture because we ourselves consider ourselves as individuals. I'm me, you're you, whatever, right? But there are lower forms in the borderland, in the boundary between one-celled organisms and multicellular organisms that are more bothersome in this respect, is the quote. And this is something that hits home for someone like me, who's a researcher in oncology, because what happens in cancer? Each of us is understanding ourselves as an individual, having that, I think you mentioned, what did you mention the word? Identity, right? Individual identity, self. And not just mentally, psychologically saying, I'm like this. You can describe yourself quite well. You can look in the mirror and you can do all the vanity, everything, right? So there's that. But even immunologically, there's the immunological self, which is protecting itself and so on. But where is that self? what is the location of that cell and what is the granularity of that cell. So if someone had to ask you, where are you literally? Yeah, okay, you're sitting on a chair in this room. But what is that cell? Is it distributed amongst all the cells in your body? If so, then does each of those cells have their own sense of self? Because bacteria clearly, bacterial cells have some sort of boundary, self, non-self, right? So do unicellular organisms, amoeba, there is a boundary, self, non-self. Clearly, the cells in the body also have that boundary. What happens in cancer?
The intercellular contract breaks down.
Exactly. Thank you. Intercellular contract breaks down, meaning rather than all of them working in unity towards preserving this identity of self of the organism, cell is sort of acting individually as a group of multicellular cells all trying to survive in their own. And so this paper goes through that body patterning, cognition, okay, from a psychological aspect. But they do hit upon this multicellularity versus cancer, the shifting boundary of self, okay, from a very different angle. And now I wanted to bring you to one of the figures. Yeah, this. So the central hypothesis, thesis of this paper is that each different sort of organism perhaps has a different concept of via which or a way of understanding the space-time aspect of their existence. So the way we humans comprehend the space-time aspect of ourselves may be very different than the way a dog or a tick or any other organism might comprehend. We're now going to go into the issue of what we mean by comprehend even, because again, yeah. But looking at it from this angle, combining this kind of in your mind with the assembly index approach to try to define life in that manner. Why are suddenly researchers focusing on this? Because you see funding going towards trying to find life out there, trying to see if humans can live on Mars or other places. So there's lots of applications for broadening your mind and thinking in these new ways. There's a need for new areas of research to open up. And with that, we shall close today's session. Hopefully it was enjoyable to all of you and you learned a little bit of something. Please do not forget to answer these questions, especially the question number 1 for yourselves. It's more like for you to have a take home from this workshop. Can you use anything we learned today to help you in your research or teaching or learning?
Okay, cool. Thank you so much. We have some time for questions. You need to ask her anything, something that she discussed today in last 2 hours or even beyond that. We'll of course have some informal interaction just outside this room where you can talk to her. But any question, anything you'd like to ask her? Anybody who has joined us on Zoom or YouTube? What you try to do is not very easy, which I realize now at the end. It was not easy. When we discussed, of course, there were a few parallel understanding that we did not discuss. But what just happened was, very kind of a multidisciplinary, process as well, where you try to get different things together. I hope individually, every one of us got something out of it. Either the way you presented this, you engaged everybody, or something that they started thinking about now because there was a discussion happening. Maybe something that they are studying right now, or they are teaching right now. So this is from a young teacher who just came here and
Wonderful. I could sense that.
because we know some of the participants here, we know where they come. But this kind of interaction is really necessary. You and Saurabh mentioned at the beginning. We all with different discipline and specialties have to come together and have such kind of interaction that you did right now. Thank you so much.
My pleasure.
Now, before that, I just quickly introduce my team. So Jasmine Dugal, she's a physicist. She works in science communication. We have Dr. Manwadi Lekar, he's a biologist. It's dealing with all the Zoom that was happening right now. We have with us Ankit. Ankit works in Science Activity Center here at Pune. So the different levels I mentioned, I'm going to come to that now. Naveen, who helped us with all the IT support, thank you so much. And we have Rahul Iyer, who was a student here at Pune and now works at Science Media Center at Pune. So this was possible because of different people coming from different groups with their own specialities, something that we did. And Rohan is here. Rohan Sankar is our backbone, our administrator. He took care of everything that was happening behind. So, thank you so much. There is one more person, Omkar, who's a mathematician. Part of our team is not here right now. And before we leave, we have Dr. Sujata Varadarajan. She is our collaborator. Yeah, our collaborator who is here for the session. Also, we'll have opportunity to talk to her. She has got some information of MSFDA, what they do and what different centers work on. So she's also available here for some time. So you can talk to her. And somebody is missing here,
Yes.
who is Dr. Neeraja Dashputre. She joined us through Zoom. She is the chemist of our group who works here at ICER. She's a PTO. She's currently presenting her work they have been doing with another project we have, what Saurabh mentioned at the beginning, for schools for the iRise project. So she's presenting her work in a chemistry conference at time. So everybody, thank you so much. Am I forgetting anything? That's the only thing I know. Okay, Mayuri, welcome.
Just to give you a very brief introduction about MSFDA, and we are Section 8 company funded by the government of Maharashtra for specifically conducting programs like this, more for the faculty development. Of course, we have programs for students as well. And ICER is one of our collaborators. We have around 40 collaborators, ranging from ICER to IPHF family or Symbiosis training or or Lalit Kala. So we all these institutions who have something unique with them for imparting to the as a training program for the faculty development, specifically in the context of NEP, which has come up for implementation. So MSFDA is basically geared towards helping faculty members understand what exactly these difficult terms in NLP means and what are the possibilities of its implementation. So that's the role of MSFDA. We have a brochure and newsletter, the driven newsletter, which Bhargav and Mayuri will be handing over to you. And please visit the website, and also you could enroll for the other programs. And feel free to contact me or anybody from my team or any contact that you get for MSFDA. And if you want any further information, you could give me in touch. Thank you. Welcome to this joining this workshop, and welcome also to the family of MSFDA, because we are also one of the objectives is to develop this, community of teachers and practitioners in the educational field, so that we are together able to do something. And thanks to Asim and his team, because they've been there with us since the very inception of MSFDA. I think the first part, they were the first collaborators with us. and they've gone a long way, and of course they contribute. And we look forward to seeing them move much faster than what they've been doing, because we have 5 lakh teachers all across Maharashtra.
Yeah, and I'd also like to say a couple of words that, first of all, a big thanks to Aseem and his team, all of you. It wouldn't have been possible without all of you. And all of you participants, in person and online, please give yourselves a big hand, everyone, because it's not possible without the participation we got today. So,
Yes, Comedy, thank you on behalf of MSLPA. So, Comedy and I were colleagues.
Colleagues. Yes. all of us,
Yeah.
Aseem and
And
Sujata,
perhaps 10, 12 years back.
10 years ago. Yeah.
So nice to see you again.
Yes. Thank
And
you.
of course, that for like this.
Thank you. Okay.
So we have tea coffee outside. So those who would like to interact with Kaumani can take something out and come back. She doesn't have much time right now, but we'll add her to the WhatsApp group so that we can continue interact. Ankit sir, Ankit sir.